All right. Welcome everyone. We are live and it looks good on my side. Welcome everyone. Today we're going to have our third lecture on PNS genomics, and we're going to be covering the overview of steps in when sequencing a genome using DNA sequencing technologies. We'll mainly be covering two, let's say, popular sequencing technologies and show them how they work in general. So I guess let's get started with recalling again the main steps in the genome analysis pipeline that we covered last week. So as you may remember, we start with a sample collection. So this sample could be, let's say, a blood sample or any particular example, like particular sample that you can collect from a surface or essentially anywhere that may contain a DNA, let's say, or RNA, other nucleic acid molecules. And essentially this, once the sample is collected, the next step is to prepare the library in a nice way so that the sequencing technologies can accurately and with a high throughput read or sequence these molecules. And the library preparation usually includes a few steps such as purification, let's say removing other particles that we are not really interested of, and also the amplification, for example, multiplying the amount of DNA fragments such that these DNA fragments can be sequenced perhaps many times using these DNA sequencing technologies. So this is mainly a VAT solution that is inserted to the sequencing technologies, and this library is essentially an input to the sequencing technologies, where this particular library includes, let's say, the chopped or fragments of DNA, let's say, to sequence. And essentially the sequencing technology takes this library as an input, and then as an output it generates some data. So that some data is not directly, let's say, ATGCs, the sequences of bases, but they first produce some raw sequencing data. So this raw sequencing data is generated based on the principles of the DNA, these sequencing technologies, where, for example, one technology may generate some series of electrical signals. Some other technology may generate, let's say, a series of images or even a movie, let's say, as a raw sequencing data. And then essentially next, what we do is that we analyze this raw sequencing data mainly in two ways. We either directly analyze this raw sequencing data to do some genome analysis, or convert this raw sequencing data into let's say, the sequences of DNA like ATGCs, which are usually referred as base called reads, and so that we can put these sequencing data in a human readable form for the later steps in genome analysis. And there are essentially many steps in genome analysis, such as base calling, that can basically translate the raw sequencing data to the sequences of bases in a human readable form into a set of base called reads. And then in the next steps, we can perform, we can use many tools such as error correction if the sequences are erroneous, such as mapping, read mapping, assembly or variant calling to identify the mutations and so on, and essentially many other steps. And since we have basically many different pipelines and many different steps, there are also a huge number of tools developed to perform each step. And this is some summary of these tools, perhaps even outdated for today. So how we ended the last lecture, it was essentially by showing this picture, which summarizes some common steps or a common pipeline in genome analysis, which starts with the DNA sequencing, goes up until variant calling as usually one of the last steps in genome analysis. And we said we're going to be focusing on the sequencing part in the next week, which is essentially this week. And this is essentially what we're going to do this week. So then let's start. So let's see how we essentially take the set of DNAs in their biological form, let's say, as molecules, which is not in a, let's say, in a digital data form or not in a human readable form, right? And then see how we can essentially use these DNA sequencing technologies to convert them into data, let's say, in a digital form, so that we can use this data to analyze in the later steps. So to start with, we're going to essentially talk about a few limitations of DNA sequencing technologies. One limitation is essentially is one of the big limitations. So we can start describing this limitation by asking the question, basically, can we sequence the complete genome in one piece? Meaning, let's say, remember, we have in human 23 pairs of chromosomes, and essentially, can we, let's say, so this is the largest, let's say, fragment that we can have in a genome, right? It's a single piece of chromosome. And the question is, can we give this particular chromosome as an input to our DNA sequencing technology and then take it again as one piece as an output? And the answer is mainly no. So there is no machine today that can deterministically take a DNA fragment, let's say, in a complete unfragmented way, like in a complete piece of as a complete piece of chromosome. There is no machine that can take this complete piece as an input and then generate again the complete piece as an output. And rather, we DNA sequencing technologies generate the fragments of DNA, because this is how they take the DNA as an input. So the goal of DNA sequencing in general is to figure out the complete sequence of ACTGs in an organism's DNA, let's say, in the case of DNA sequencing. And the challenge, as we said, there is no machine that takes the long DNA, the complete DNA in its complete form, let's say as a single piece of chromosome, as an input, and then gives the complete sequence as output. So this means that what we sequence is actually the chopped or the fragmented pieces of DNA, and essentially as an output, what we get is again the fragments of DNA where we don't know essentially where this DNA is sequenced from in the original genome. So we lack the order information and we lack the position information after sequencing. So essentially, DNA sequencer is a chopper. Let's say it's not particularly itself is chopping the DNA, but since it requires, let's say, at a certain level, the chopped DNA, but how we prepare the libraries usually. So we have a huge one piece of, let's say, a chromosome, but then essentially we, in our library preparation, the DNA is actually fragmented into small pieces, and then these small pieces are then sequenced to construct the sequences of bases that we call reads. So these fragments of DNA are sequenced and then converted into human readable form, which are known as reads. So this means that we have this bunch of lots of reads that we don't know where they belong to in the genome. So the goal is essentially to figure out where they belong to, right, in the genome, so that we can reconstruct somehow the individual's genome, usually by a step known as read mapping. And this is essentially a costly process because of this main limitation of DNA sequencers. So there are essentially a large number of DNA sequencers today. So these three sequencing technologies, or the companies, are essentially the main market leaders. One is an Oxford Nanopore Technologies, the other one is PacBio, and the other one is Illumina. And there are many more, essentially, the sequencing technologies today. And of course, they offer different trade-offs between them. So, for example, these are some machines from Illumina, and they have essentially different runtimes. They have different read lengths, but if you look closely, these are usually in the range of a few hundred bases, right? So these machines can generate from 100 to somewhat 200 or even 500 and so on read lengths. And essentially, their sizes are also different. So this can be a huge, let's say, a sequencer, or like a relatively small sequencer that you can put on your, let's say, a small desk. And of course, their prices differ based on their capabilities and so on. So this is an example. These are some Nanopore sequencers. So if you are following, let's say, the Apple events closely, you might have, let's say, observed one of these actually in the recent Apple event that happened 2 days ago, which was one of the Promethean machines sitting on a desk next to the new, let's say, MacBook Pros, which we're going to be covering, let's say, the benefits of these Nanopore sequencing technologies soon. But I wanted to mention that maybe you already have seen one of these machines, but you're not aware of, let's say. So essentially, the Nanopore sequencer technologies also have different, let's say, machines or devices in different categories. Some of them are very small, some of them are relatively large, and then their sequencing prices varies a lot, and also essentially how many bases that they can sequence in a single run also varies. Essentially, there are nowadays newer sequencing technologies producing or providing lots of, let's say, benefits and lots of opportunities. So if you're interested in learning about these opportunities, you can take a look at this paper. But as I said, these different DNA sequencing technologies generate different raw sequencing data. As we said, for example, Illumina machine generates, let's say, a set of images to represent, let's say, a different bases at different positions. And Nanopore sequencing usually generates raw electrical signals, and PacBio generates, let's say, a series of images which is in a movie format. But again, they have different trade-offs. Some may be generating short reads, but they can be accurate. Some others may be generating long reads, but they can be slightly less accurate and so on. So we're going to be covering these trade-offs. But let's start with, let's say, one of the sequencing technologies, which is Nanopore sequencing. So Nanopore sequencing is one of the widely used sequencing technology in the field. And there are certain advantages. For example, it can sequence essentially ultra long reads, not even long reads, but ultra long reads up to a single a read can be up to 2 million bases, which is actually huge compared to the other existing sequencing technologies. It also offers some benefits, relatively unique benefits, such as these machines can be really small, like you can even like hold it in your hand, such that this can offer some portable sequencing advantages. So you can carry it with you, even connect it to your computer, personal computer, and then sequence it on the go, let's say. And these are relatively cost effective, and there are also some unique features such as real-time analysis that we're going to be covering now. So let's look at how this nanopore sequencing works and what is real-time analysis, essentially. So here, imagine that there is a three-layer. So this is essentially a three-layer field. Imagine we have a top layer, we have a middle layer, and a bottom layer over here. And the top layer, and on the middle layer, we essentially apply a voltage in a certain direction, such that the top layer is negatively charged and the bottom layer is positively charged. And essentially what happens is that the DNA, the nucleic acid molecules, the DNA, for example, is initially located on the top layer. That the DNA is the DNA fragments that we want to sequence. And since the DNA is also negatively charged, what happens is that due to the electromagnetic field, the DNA over here essentially moves towards the positively charged site, just because how we apply the voltage, essentially. And essentially, as it moves, there is a tiny pore over here located that we call nanopore. And what happens is that this particular DNA, as it moves towards this positively charged site, it essentially attaches to this nanopore and then continues its move. But essentially what happens is that as it moves, what we do, or what nanopore sequencing does, is that the ionic current changes are measured as it moves essentially through this nanopore. And this ionic current measurements are significant enough such that we can tell what sort of base, or what sort of, let's say, nucleic base or character is being sequenced at a time. Because this sampling is so fast that it happens around 8 to 10 times per base. So what this means is that we are generating electrical signals because we're observing the ionic current, we're measuring the ionic current changes as the DNA moves through this pore. And what we're doing is that we're generating around 8 to 10 current samples per base over here. And this particular raw data, which is in the electrical signal form, is generated continuously as the DNA moves towards the negatively charged side to the positively charged side. And essentially, this nanopore sequencing idea has been around, let's say, since 1980s. And what I mean by the idea is that the fact that the DNA is negatively charged, and the fact that actually we can somehow, let's say, understand each base from the ionic current changes that they would make as they move from the negatively charged to the positively charged side. But the problem back then was the speed, let's say, that we would not be able to control the speed of this DNA moving towards this negative charge to the positive charge. But this means that the DNA, essentially, if DNA moves in an extremely fast way from here to here, and essentially your sampling mechanism, if it is not fast enough to catch the change for every base, let's say, then we're not really sequencing the DNA for every single nucleotide, right? So we'll perhaps even be sequencing it for some random bases where there are some gaps between these bases. So the real challenge was to control, let's say, the speed of this DNA moving towards here to here, and then this speed is essentially controlled by this particular protein over here on top of this nanopore, which is essentially the trademark, let's say, of some companies such as the Oxford Nanopore technologies, which is let's say the trade secret of that particular company, so that they have an advantage, let's say, competitive advantage to make it profitable. But as I said, the nanopore sequencing idea has been around for the last 4 decades. So then I guess we can cover the next step in nanopore sequencing, which is essentially understanding this raw electrical signal. So as I said, these raw electrical signals are significant enough so that we can understand each base in the DNA. And to understand this, there have been several mechanisms developed over the last 10 years, maybe even more than that. Initially, we started with some, let's say, naive or basic statistical methods, such as hidden Markov model-based mechanisms, and then recently we moved towards machine learning models that use, let's say, deep neural network that essentially understands from this raw signal and then translates this into the set of bases that we call a base calling process. And essentially these would be called base called reads. So this is what base calling is: translating the raw signals, raw data in general. So if you're talking about nanopore signals, yes, these are raw electrical signals. But if you're talking about like any other sequencing technology, they're going to produce raw data, and that raw data needs to be translated, and this step is usually generally referred to as base calling. But there is actually one more feature, the unique feature that nanopore sequencing is providing, which is the real-time decisions that we can make during sequencing. And these real-time decisions are made based on the real-time analysis. So what is real-time analysis? Real-time analysis, this raw electrical signal is generated in real time at a certain throughput. So the throughput is usually around 5,000 current samples per second for each nanopore here. So this means that if there is a computational mechanism that can analyze all these 5,000 current samples within a second, then this means that you can do your analysis in real time because you're matching the throughput of nanopore essentially. And what we can do with this analysis is assume that you have a sample where you have organisms from a human, from, let's say, a virus, from some other organisms, but essentially you're interested in, let's say, understanding whether that particular sample is contaminated with a particular virus, let's say. For example, we're doing COVID tests, and we want to understand whether there are a large amount of reads coming from COVID genome. So what we can do is that we can extract all these signals and then somehow try to understand whether that signal is coming from a COVID genome or not. And if they are not, let's say, coming from a human COVID genome, or if they are coming from a COVID genome, as soon as we basically find an answer to this question, what we can do is that we can tell the sequencer to stop sequencing that particular read while it is not sequenced fully, let's say. So in the middle, we can tell the sequencer, all right, we don't want to sequence that read anymore because now we found an answer to our question. For example, this read is coming from a COVID or not, and I don't want to sequence it anymore because this is just a waste of time and waste of resources for me. So we can, based on this real-time analysis, we can tell the nanopore sequencer there is such a functionality. And how this works is relatively quite easy. So you can, whenever you want to stop sequencing that read, this means that actually you want to reverse the direction of sequencing. You want to eject. So you were inserting the DNA towards the negatively charged or the positively charged in the initial step. But what you want to do is that now you want to reverse the direction of sequencing so that you can eject that read, and then so that you can stop sequencing. And then how this works is that what you do, you literally reverse the direction of voltage here, such that now this part is positively charged and this part is negatively charged, such that this DNA now moves again towards the positively charged, but now the positively charged side is here, essentially. And this is how stopping the sequencing works. And of course, there are some challenges to meet to make this effective and efficient, let's say. And essentially, since we are excited about the opportunities that the Nanopore sequencing provides, we also publish several works, such as RowHash. So RowHash essentially analyzes the raw signals in real time and makes these decisions in real time as well, so that we can effectively and efficiently use Nanopore sequencing for these real-time decisions. So if you are interested in learning more about this field, you can take a look at this paper. And we are also improving our tools, our algorithms continuously. So this is the next work that we recently put on archive, so you can take a look at this as well. So this is another work that builds on top of RowHash again, and what it does, it performs approximate string search, let's say, or approximate signal search by doing some alignment using some clever algorithms to make the analysis even more accurate. So if you're interested, again, you can take a look at this paper. And we're also interested in making the base calling process faster by reducing its, let's say, workload by some quick understanding of these unnecessary works that are usually sent to base calling, but we quickly analyze or quickly identify these unnecessary works, and then so that we don't send them to base calling, so that the overall base calling process is faster, let's say. And these Nanopore sequencing devices are relatively cheap, so you can buy a single flow cell around $900 US, and with this single flow cell, you can actually sequence your entire genome more than once, usually twice or even 3 times, depending on, of course, like how well you prepare your library. But essentially, with a single flow cell, you would be able to analyze your entire genome in an effective and fast way. And we're always talking about these ATGCs, but actually a DNA molecule, the nucleic acid molecule, contains more information than just nucleic bases, based on their, let's say, chemical structures, based on the other, let's say, chemical structures that bind to these nucleic molecules. And understanding those differences is also very important for many reasons. And since raw signal data contains this information, there is this paper discussing that how we can essentially utilize this extra information, meaning the information other than just ATGCs, how we can utilize this extra information in raw sequencing data using machine learning techniques. So this is a very interesting read, so you can take a look at this as well. So this was how Nanopore sequencing works. So let's take a look at the next, the other sequencing technology that is used in Illumina machines. So Illumina is a company that utilizes a particular sequencing technology that is known as sequencing by synthesis, or SBS. So here, essentially, you can assume that this is not in a human readable form. This is essentially the nucleic acid molecule, and the molecule is essentially a set of ATGCs, right? And we want to essentially sequence this to translate this into a human readable form in sequencing. So, and of course, we know that A binds to T and G binds to C due to the chemical bonds, let's say, between them. So what happens is that in SPS, in sequencing by synthesis, so there are although like several ways of doing it. I'm going to try to describe one of the common ways. So what happens? So what happens in SPS is that since we know essentially this binding between the A and T and G and C, you can assume that this particular molecule is attached to some surface. Let's say it's literally sitting here like this, and then what we do is that we insert a bunch of ACTGs around it. So there are a bunch of ACTGs, like in terms of nucleic bases, floating in a free form. They are everywhere, essentially looking someplace to attach. Right? This is a single T. So what happens is that, so once, for example, all of these ACTGs are floating around, assume that now we have a T somewhere here. And what happens is that since T binds to A, since we know that, what happens is essentially as soon as it binds, there is some chemical reaction, there is some clever, let's say, design behind this process. But as soon as this T binds to A, there is let's say, a light that is being emitted at a certain frequency. This certain frequency is very important because as soon as the light, the fluorescent essentially being released at a certain frequency, we take a picture of that emission. And since essentially we know which frequency corresponds to which base, we can tell based on the frequency that the T just bound to a particular position in the DNA. And since we assume that DNA can bind to A, right? So we tell that, okay, at this particular position here, at this position, there should be an A. So this is essentially how we sequence using sequencing by synthesis. And you should assume that there is not only T, but there are a bunch of other bases, right? The ACTGs floating around these copies of DNA fragments. So this is this surface that I was mentioning you, and there are many copies of this particular DNA fragment. So assume that this is the DNA fragment that we're sequencing at a certain time. And what we have in reality is that there are many copies of the same DNA attached to the nearby positions, let's say in the same cluster, let's say. And there are many ACTGs in the free form floating around. And what's happening is that they are binding to the next available position in the DNA. And then we're continuously taking pictures to generate the images of this process. So one question that you may ask, since these ACTGs are in a free form, how can we essentially ensure that this, for example, this T, for example, binds here, but doesn't bind here, let's say? Or how come all these ACTGs are not binding at the same time to different places, right? Because then it would make our sequencing very hard, because what we really want to do is that we want to do it cycle by cycle, base by base, essentially. So we would, for example, expect this T first, let's say, binds to something, and then once the sequencing of this particular base is finished, now we want, in the next cycle, we want to go next, and then we want to sequence this G now, not, for example, at the same time this A and G, because these bases can be binding to many places at the same time. So again, this is solved by some clever decisions in sequencing. What happens is that you may imagine, let's say, there is some imaginary bond here attached to or nearby to the base that we are supposed to sequence next. And what happens is that these bases can only attach to a base only if there is this particular bond being placed nearby the next basically base that we are supposed to sequence. And if there is no such bond, for example, in the middle, there is no such bond that these bases can bind to, and that's why essentially these bases cannot bind here, but binds somewhere that there is this particular bond. And as soon as this T is bound here, then this particular bond washed away, and then for the next cycle, it essentially binds to this, let's say, next base, so that now we can sequence this next particular base in the DNA sequencing. So this is how SPS is working, and this is done at every cycle, base by base. But essentially, the problem is that we cannot do it for so many bases. We can only do it at around, let's say, 400 to 500 bases, and this is basically what's limiting the read length in Illumina technologies. If you, by theory, by practice, you can actually sequence reads with 1,000 lengths, a few thousand lengths. But what happens is that since we're sequencing the copies of the same DNA, these DNA should be in sync, meaning on the surface, let's say, if we're sequencing the bases on the surface at a time, on the surface, we should be seeing the same base all the time. But this, because we're taking the picture, and what we want to see is that we want to see the same, let's say, color every time at different places. But in theory, in practice, this is not the case, and some reads go out of sync. Meaning, on the surface, for example, we may have different bases, just because this particular read is going out of sync across cycles. And this going out of synchronization becomes more serious as we go towards go with further cycles. Essentially, if you are at, for example, 500 cycle, perhaps you can assume that almost all the reads are now out of sync. So this means that whatever you're generating here is some random bunch of images. Then this image will not really tell you something very informative. So the critical thing is to finish the sequencing before many of these reads go out of sync, such that you can do your base calling from these images accurately. So this is essentially the limitation in the Illumina sequencing, and you can take a look at this link to learn more about Illumina sequencing. But again, all of these reads, regardless like whether they generate short reads, which are around a few hundred bases, or longer reads, which can be around few thousand bases, or even up to million bases, still they are the fragments of a larger piece, for example, a full chromosome. So this means that regardless of the sequencing machine, these reads still lack the information about their order and location, because they are DNA fragments, right? We chop them. As soon as we chop them, they freely float around your, let's say, vet solution, which is your library, and they don't contain the information about their order, in essentially where they are chopped from in the chromosome, right? They won't tell us, I was chopped from chromosome, let's say, something at position here. If you had this information, our life would be extremely easy, but we don't have this information. But what we rather have is just a bunch of fragments of DNA coming from somewhere in the genome where we don't know which position, and our goal is to figure that out. This is essentially one big problem. We need to construct the entire genome, let's say, if you're doing whole genome sequencing, from many sequenced reads. So this is like solving a puzzle, right? So we may have these pieces, right, which the fragments of your DNA, which are reads, and our goal is to solve the puzzle by putting these pieces together to reconstruct the picture again. And one good, perhaps, news is that we may have, let's say, a hint that we can look at, which is known as the reference genome, which is almost a complete genome and a representative of a certain species, such as human reference genome, right? So we mentioned this in the last lecture that we finally generated the human reference genome in one piece. But since each individual is different from each other, we cannot really essentially just use that reference genome for every analysis. We also want to understand, let's say, the mutations, or we may have some sequencing errors, right? So there can be some differences between the image that we're trying to construct and the reference genome. But still, this can be a good enough hint where we can look at this picture and then try to figure out where each piece may belong to in the picture that we're trying to construct. And there are, of course, some trade-offs between sequencing technologies that's making this reconstruction of the picture either challenging or easier. For example, if you have Illumina reads, what you end up getting is smaller pieces compared to the other technologies such as PacBio or ONT, where these technologies are producing relatively longer reads. So this means that when you have short pieces and solving a puzzle, it's relatively more challenging to solve that puzzle, because you may have a piece where you may not know whether that piece is coming from here or here, because they may be extremely similar to each other, maybe can be exactly the same. And you may need to make some random decisions here. And once you make those random decisions, it may affect the further downstream analysis. And I forgot to put this paper here, but we have a paper about this. Once you make these random decisions, this is actually affecting your further downstream analysis. And the paper is from 2016, and the title is on genomic repeats and reproducibility by myself, Can Firtina and Janal Khan, which was published in Bioinformatics. So you can take a look at this paper to learn basically how these random decisions are affecting the further analysis, the analysis in the further steps in the genome analysis. But again, then I guess if we just talk about the length of the reads, then maybe the winner is clear, right? So why not just use these longer pieces, because it's easier to construct. But then the other problem is these longer reads usually have higher error rates. So this is now actually nowadays around 5% to 1% range, not 15%, but this used to be the case a few years ago. So you can see basically the trend in the development, in the accuracy improvement in these long sequencing technologies. And these short reads have pretty low error rates, almost negligible. So this means that whatever you're constructing using these long and erroneous reads, you may construct something, but it may not be the perfect image. You may essentially end up getting some an image that includes some errors in it. So there are essentially these trade-offs, and this is the picture that I showed you earlier. So this is perhaps the trade-off chart that you can observe here. So this is essentially, these are the two, let's say, the ideas or technologies behind in DNA sequencing. We covered the Illumina, the sequencing idea behind Illumina, and the sequencing idea behind Nanopore sequencing. We didn't cover PacBio, but this is relatively similar to sequencing by synthesis used in Illumina, but rather actually does the sequencing by attaching a certain enzyme to the DNA strand, and again, the free form, let's say the bases are attached to the DNA, but then this attachment happens not cycle by cycle in a very continuous way, and such that the movie is generated rather than the images that are taken cycle by cycle. And this way, PacBio can produce longer reads with a slightly less accuracy compared to Illumina. So in the next step, we're going to be focusing on read mapping, which essentially is somewhat solving life's puzzle from sequencing output, where we're going to be looking at the algorithms used for figuring out where each DNA fragment or where each read may be coming from in the genome itself. And we're going to be seeing essentially what the algorithms are, what the data structures are, to make it practical, to make this analysis faster, let's say. So this is the topic in next week. So with that, I'll finish the lecture for this week, and I'll see you next week.